We can move to the next speaker, who is Dmitri Simberg, an associate professor at the University of Colorado. And today he's going to present a talk on the mechanism regulating accumulation of nanocarriers in skin and the relation to toxicity.
Thank you. Can you hear me? Yes. Thank you very much. So again, this is our campus in Colorado. So thanks, Tori, for the nice introduction about the caveats of imaging and the labeling of things. I'm definitely aware of these caveats. So and my interest in really the barriers, how nanoparticles accumulate in tumors and immune cells, and what kind of governs the biodistribution. So this kind of research was very much aligned with my goals. And the first phenomenon of skin accumulation we observed when we built a near-infrared system to basically kind of fiber optic system to image nanoparticle accumulation in tumors. So we kind of attach it to the tumors, but we actually see that most of the stuff really builds up very quickly. You know, here's fluorescent dextran and stays after things are cleared from the circulation. So, and we realize that things are accumulating in the skin, and we're just basically imaging the skin that's over the tumor rather than the tumor itself. To better understand this problem, we use the intravital microscopy, where you kind of create a skin flap in the mouse belly, and you put a microscope, and mouse is breathing, so it's really inconvenient system, but you can inject nanoparticle and see at different time points what happens. And there is red are liposomes, and dextran labeled blood vessels and host nuclei, and you can do it in a living mouse. And you can see that at 5 minutes there is binding of liposomes, but really quickly they start getting out of the blood vessels, and you can see them accumulating in the all kinds of dermal cells in the skin. And 7 days after the injection, you can still see the fluorescent label accumulating inside the skin. So we switched from the intravital model because it was not very convenient. So we actually realized we can just cut out a piece of skin at different time points and just put it directly on the cover slip and look at the whole skin with a confocal microscope. And that gave us really unprecedented resolution for the imaging. But before we did that, we kind of just wanted to see where the liposomes are going when we inject into mice. And we label with DIR, liposomes with DIR, and we look at different organs. And what we see that there's skin accumulation, it's all over the skin, but there are some hot areas. And these are hot areas which are in the feet, in the foot pads. This is prominent accumulation, and also in the spine. You can see where the spine touches the skin, and also where the ribs touching the skin, suggesting that really the areas of pressure accumulate a lot more signal than the rest of the skin. And this accumulation is as high as in the spleen, which was pretty remarkable to us. Like, it's not known that skin accumulates that much. But we kind of started to understand this problem a little more, realized that many drugs show toxicity, and Doxil is one of them, pretty notorious for this called PPE, or hand-foot syndrome, where the areas of pressure are actually showing this ulceration, severe pain, debilitating actually thing. In 25% of Doxil patients, they may require a dose, a modulation, or even discontinuation. But actually other drugs do that too. Multikinase inhibitors, 5-FU. So it's really an important problem we started to identify further. But what's known about how nanoparticles accumulate in the skin? And actually the only thing that we can find in the literature is that the hydrophilic coating of liposomes appears to cause drug accumulation. So not really a lot, So I think we can do better than this, okay? If we really want to understand this problem and try to deal with solutions, so we have to really know more about what happens. If we inject Doxil, and Doxil is really a long circulating drug, in mice it's about 30 hours half-life, we inject Doxil and then look at the accumulation of Doxil at different time points, and we see that as soon as 1 hour, we start seeing accumulation in sweat glands or sebaceous ducts, and some hair follicles too. There's some chelation in other organs, areas of the skin too, but at 1 week, we can see doxil over the place and the sebaceous gland and hair follicles. So it looks like it started accumulating pretty quickly in the liposome. So it's 1 hour accumulation. That's what decided as our time point to look at the accumulation of the liposomes. So here we injected the ID-labeled liposome, and we used that skin flap to do the confocal microscopy and the white blood vessels. And what you can see is really kind of a cloud that coming out of the blood vessels. It was really striking how we see this sort of like particles sort of accumulating. We can quantify the by segmentation of the image. We can quantify how much is extravascular, how much is intravascular. We see about majority of them is actually extravasating, get out of the blood vessels. So, but what are these particles really? These tiny little particles? We didn't have a clue about that. We thought these are maybe exosomes of some sort, or particles. So we, to understand this problem, we did a sectioning, fine sectioning of the pieces of skin, and really did a high magnification confocal microscopy to really see. And here we labeled the liposome with 2 colors. So we just put 2 membrane dyes of different colors in the membrane to kind of see where the liposomes are. And we see these are colocalizing, nicely colocalizing liposomes. And in the skin, as soon as in 1 hour, we see blood vessels, and we see the particles of 2 colors, both colors, which suggests these are probably liposomes, kind of like we injected them as 2-colored particles, and they come out of the blood vessels as 2-colored particles. So here is a different one where we label the membrane with the, 1 type of color, and we put a rhodamine, red color, inside liposomes, and they look like they're double labeled too. And you see the same double labeled particles coming out of the blood vessels. It looks like almost intact particles are getting out of the blood vessels, but you also see some smear of the fluorescence. It looks like there's like 2 modes of extravasation. One is like individual particles, liposomes, and the other is diffusion of the dye from the blood vessels. Here we thought, okay, maybe these are liposomes, they kind of breaking apart, so we use a solid iron oxide nanoparticles called nanoworm, labeled with Psi3, mixed with the liposomes. They're more or less similar size, and what we see that both liposomes and iron oxide nanoparticle getting out of the blood vessels. So it looks like there is strong evidence that nanoparticle can really diffuse out of the blood vessels intact. With the liposomes, though, you can see those smearing. That means that the lipid dye kind of migrates independently, but some liposomes definitely getting out of the blood vessels independently. Here we use large non-extruded liposomes together with these iron oxide nanoparticles, and you can see that there is still this diffusion of the green dye, the ID, but no big liposomes getting out of the blood vessels, but you still see iron oxide nanoparticles getting out of the blood. So it looks like there is some size threshold by which like liposomes kind of can get out of the blood vessels. So how things can extravasate? What do we know about extravasation? One is active transcytosis, of course, that was shown by Warren Chan. There is passive diffusion via gaps, and what we always think about what happens in the tumors. There is some extracellular vesicle transport. Maybe the particles kind of be part of exosome biogenesis and get in tissues. And there's leukocyte trafficking. So how we can really understand what's going on? Looks like we have something like an intact liposomes, but really don't know still how they pass through the skin capillaries. So first we stain for the extracellular vesicles for exosomes. There is no colocalization between exosomes. There are exosomes actually in the tissue. They seem to be getting out of the blood vessels. These are red dots. Like CD81 staining, but looks like the liposomes are not really colocalizing with them. Not exosome, but this is not the ultimate proof. And then there was this experiment that Warren Chan did in one of his papers, where he just described something called a zombie model, where he can inject nanoparticles with formalin, or excuse me, nanoparticles, inject mouse with formalin. You perfuse mouth with formalin, everything is dead. But if there are pores and gaps, they're still preserved, and the particles can diffuse. It's really an elegant really very brilliant things for him to do. So we just did this thing and we perfused mice for now 1 hour in the perfusion apparatus with liposomes, either fixed mouse or non-fixed mouse. And here is that skin that food skin that we excise. And what we see that in both cases, we see this extravasation. We see both lipid diffusion and the individual liposome extravasation, almost like the fixation didn't do anything. So this is, I think, the ultimate proof that what happens is that liposomes or nanoparticles extravasate in the skin not through active transport, doesn't require any energy and any leukocyte uptake. We've been perfusing just with the perfusion buffer. There's no leukocytes there. So that kind of like a strong, so in last kind of evidence that we did TEM of the skin injected with liposomes, and you can appreciate those vesicles. This is red blood cells. These are vesicles, probably liposomes, and you see a gap between the endothelia, kind of like, like not adjacent. You don't form the tight junction. So it's very interesting how in the skin. And I think the skin is interesting in that regard because it's a very loose tissue. It's not like a tight tissue. So you can look at the transport in the skin and probably make an interesting conclusions. So our idea is how this happens. First, there is a passive transport through some sort of gaps, that probably pre-existing gaps in the skin in the areas of pressure. And second is through some diffusion of the lipid from the endothelium. But what happens to the mice or animals when we inject Doxil, really? So here's what you can do. If you inject them several times a week with Doxil, and you can get some sort of toxicity. It's not really PPE, but it's something that we can work with because it's necrosis of the skin, not similar to people's toxicity. Okay, but it looks like there's some sort of inflammation, but we don't actually see a lot of inflammation. We see some COX-2 overexpression. We don't see an obvious like immune infiltrate or something. We see some vacuolization, almost like a tissue is dying. Not much inflammation marker, actually. No classic inflammation. That's what actually they say in the literature, in the patients, when they did the biopsy. There's not much inflammation going on. There is something else going on. But what I'm leading to next, what happens really paradoxically, when you inject a mouse with Doxil, or rat, in this case in the rat, you see after the accumulation, it kind of opens the gate. So if you inject a second dose of the ID-labeled liposome, you see a ton more accumulation. You know, I don't think if I include the quantification here, but it's about 10 to 20 times more of the liposome accumulation compared to the non-treated. You know, we can see it in the rat. This is kind of toxicity that is shown in the rat. It's more similar to what we see in humans. And on the upper panel, you see that liposomes do accumulate in the skin. But if injected with Doxil, there is a huge increase in the accumulation of Doxil, of liposomes. So there is some opening of this. You can actually achieve that just by injecting subcutaneously doxorubicin or intradermally in a mouse. You wait a couple of days, and then inject diar liposomes, whatever labeled liposomes, and you can see really massive accumulation of liposomes compared to the irido. Again, the accumulation is everywhere in the skin, but where you injected doxorubicin, is a huge accumulation compared to this. So we kind of come up with the hypothesis, at least, of what happens. So we inject doxorubicin. There is enhanced permeability in the feet or in the areas of pressure. That's why you get accumulation in the first place. And then what happens next, we don't know, really, honestly. We try to do transcriptomics, try to understand what happens to different other inflammatory markers. I talked to Marina about that. And then there is increased permeability and then enhanced accumulation. And then doxorubicin kind of like almost recruited itself to more and more things. So the one actually asking what happens with the tumor. So I don't know, maybe that's the same thing happens in the tumors where there is some sort of not a vicious cycle that work in our favor in that case. So areas of pressure, including folk skin, show enhanced permeability. But accumulation, we believe, is not to dilute leukocyte platelet transport to active transcytosis. And initial docs start vicious cycles, but there's a lot of questions that we're trying to answer. How does pressure promote extravasation in the first place? Can we inhibit that extravasation? You know, is it possible by changing the formulation, for example, that has been trying to do that for quite some time. And does extravasation Doxil correlate with hand-foot syndrome in patient? So a lot of patients, and I'd like to thank you very much. And I stand with Israel and Ukraine in their fight. Thank you.